Good morning, everyone. Yeah, so as Chris mentioned, I'm going to be talking on behalf of Priyam, who just gave birth a couple of days ago. She's well, baby's well, and I promise I will try to do my best to describe her fantastic work that she's done, which is going to be on understanding sort of rhythmic changes in neural activity in the subsets of these cells in the SCN, which Samir had described a little bit. Okay, so this is the SCN. So this is a very simple sort of circuit, and as Samir touched on, for example, we get light inputs which are detected in the retina. You get these projections to the SCN, and this drives a variety of behaviors, as Samir mentioned, for example, sleep-wake, cycle, temperature, feeding, and so on, okay? And this is a simple cartoon showing sort of connections. So you get light detected in the photoreceptors, the IBGRCs, and then these go into the SCN, but also now we know different areas of the brain to regulate mood and so on. And then this leads to sort of biological rhythms, body rhythms, okay? That allows us to adapt to our 24-hour environment. And yeah, so this shows our body cycles through a cycle in a 24-hour cycle. So for example, during the day, you're more alert, your blood pressure is higher, your cognitive abilities are better, whereas at night, for example, your body temperature goes down, melatonin secretion goes up just before you go to sleep, and so on. And actually, this is interesting for, now you might have heard of chronotherapeutics, for example, where people use treatment based on time of day. One example being treatment for high blood pressure, right? People would give medications at night, but now it makes more sense. Your blood pressure goes up in the daytime, higher, right? So someone with high blood pressure, you give them the medication when it's higher, so it lowers down, okay? So, and this is a very simple cartoon of the cellular clock. So it's a translational, transcriptional, translational feedback loop. So this is the molecular clock system in the SCN, but you also have this outside of the SCN, right? Just to walk you through it very briefly for those who are not familiar. So we have, these are the core clock genes and proteins, clock and BMAL, for example. They're transcription factors for cryptochrome and period. These drive transcription for these genes, right? Cry, period. These proteins dimerize in the cytoplasm. They're phosphorylated by a variety of kinases, okay? And then phosphorylation leads to them translocating back into the nucleus, where they then inhibit their own transcription. So this is this clock, right? This, the basic outlay of the molecular clock in the circadian system. And well, just for a note, I'm happy to talk more about this later. This phosphorylation adds in the delay in the clock, right? So this gives you this sort of rhythmic cycling that this process goes through. And this cycling leads to changes in, for example, in neural excitability within the SCN, right? So how are these clock rhythms in the SCN, then how is this information transmitted to the rest of the body, for example, right? Or parts of your brain. So you have changes in basically firing and excitability in these SCN cells that then affects downstream targets. So typically, in the day, SCN cells are more excitable overall, okay? So the resting membrane state is more depolarized in general, whereas at night, they're less excitable, right? So not completely silent. So during the day, cells are more depolarized. They have higher, for those who are interested in electrophysiology, higher sodium conductance and lower potassium conductance. And then it's reversed at night, okay? But of course now, and then we have some data, I'm not going to show all of that, where as we go through and identify subsets of cells, these things are not exactly always true. It's more heterogeneous in the SCN. Right, and there's a bit of history. So biological clocks are around us, right? So it's, I think Samir you would have got a hint of it from Samir's talk. For example, if I recall, the first description of something what we now know as circadian rhythms was in this part of the world. A couple of 1,000 so years ago, maybe a bit longer, where I think a general of Alexander the Great, or maybe someone like that, who had described these plants that would open and close day and night. And like you see in mimosa plants, right? So you put them in the light and dark, they open in the day, close at night. And what is interesting, and this is why it's circadian, you put them in a box, remove the light, they will still go through this behavior, right? So this is a key point about circadian system. It prepares your body, right, for the next phase in a 24-hour cycle. Priam put this picture of Ron Konopka and Seymour Benzer. So we just celebrated a couple of years ago, 50th anniversary of their seminal paper where Ron and Seymour Benzer had described basically 3 types of mutations in Drosophila that impacted their rhythms, right, their period. And this caused, if I understand correctly, quite a bit of controversy because people said, how can just having simple mutations change behavioral rhythms, okay? And there was a video, but I don't think it will play on this computer, so I'll just go through. So yeah, and as Samir actually mentioned as well, clocks are everywhere. So even cyanobacteria, they have their own clock system, right? Yeah. So, there is a video. So Priya wanted me to show you this. So this is, okay, maybe not, the audio is not working. No, okay, fine. And so in 2017, Jeffrey Hall, Michael Rauschbach, and Michael Young won the Nobel Prize for their work, the seminal work on describing the mechanisms of the cellular clock in Drosophila, which is very exciting for us related in the circadian field. And Samir also mentioned, there's a lot of, so our body, we have this clock that allows us to adapt to our 24-hour rhythms. Now, if you mess around with that clock, and Samir has done some really cool work, showing that this can affect mood, right? And then there's also quite a bit of now clinical work, anecdotal work, and clinical work showing people who go to shift work or just have irregular sleep, jet lag, and some of you are suffering from jet lag. All right, this can disrupt, and this can have negative consequences in various aspects of health. So something that we're interested in our lab too is mood disorders and stress responses, so these areas, but then metabolism, so the processes in the gut are affected, metabolism is impacted, so a whole variety of things, ability to recover from injuries impacted, so healing and so on. So it shows like it's very, the normal bodily function is highly dependent on normal rhythm, okay? All right, so now going into a little bit of what we do. So we use this transgenic mice, and this follows up from some work I'll talk about in the next slide, where we use these transgenic mice we got from Doug Mahone, whose group had developed these mice where the EGFP fluorescent protein reports expression of the PER1 gene, okay? So basically, and what you notice is it's destabilized EGFP, right? So when PER expression goes up, EGFP expression goes up as well, but then it doesn't stay up all the time. It's broken down by ornithine decarboxylase, breaks it down over time. So what you can essentially get is when PER is expressed, EGFP expression goes up, and then over time it goes down, right? And as I told you, in the clock, the clock system has this rhythmic expression profile, okay? And this just shows EGFP expression in the SCN. Now, the reason Priam got into this work was coming mainly from this paper. And this is after Justin Blau, who is a professor in New York, Chris had mentioned. We were discussing this work by Mino Bell, very cool piece of paper, but it was also a bit controversial because Mino, when they did recordings in the SCN using these transgenic mice, where you have mice that express EGFP and is supposedly reporting PER expression, and then the non-EGFP, which at that time they said was not expressing PER, although it's a little bit more complicated, like always. But what they found was this. So I'll just quickly walk through, you guys, if you're not familiar. So this is over time. So Zeitgeber time is just a way in circadian biology to sort of standardize time. So Zeitgeber, ZT0 is just lights on. ZT1 is 1 hour after lights on, and so on, okay? And green is EGFP expressing cells, so these would be having rhythmic expression of the period gene. And these non-EGFP expressing cells in the paper they reported were not expressing PER, okay? But now we know it's a little bit more complicated than that. But what was interesting is Mino found, okay, so you get this rhythmic profile. So I mentioned generally the cells are more excitable in the day, less excitable at night. So ZT0 to 12 would be lights on. This is 12-hour light, 12-hour light, 12-hour dark, right? So you see these cells generally have high excitability day, lower excitability night. But Mina will also reported that within the day, for example, for here ZT2, the 2 hours after lights on, and ZT6, for example, these EGFP cells have very different states, right? You'll see here at ZT6, the cells are extremely depolarized and not really firing, whereas at ZT2, they're more hyperpolarized, relatively speaking, and have this very nice rhythmic firing. And this is odd. The reason why I say this is controversial is because typically, if a cell at this resting state, typically you think they're not healthy, right? It's very depolarized, and cells don't deal well. I mean, I know when I was working in the VTA, we're using dopamine cells, minus 25 millivolts, it's either your patch is not good or your cell's dying, okay? But you can hyperpolarize the cells, ramp them down, and you'll see they'll fire beautifully. And we've done this. Priyam didn't show any of that data of her own, but she replicated some of these findings from Mino's paper. And you see, for example, so here ZT2, at ZT6 here, the non-PERS expressing cells, they have this very nice rhythmic firing, whereas the EGFP expressing cells don't, okay? So you see the different physiological characteristics, right? So what's happening? So this is where Priyam came in, and she was interested in understanding the molecular mechanisms that may explain this sort of electrophysiological differences between these two subpopulations of cells, okay? So for that, what she did is looked at first the transcriptome, right, of the SCN cells in these transgenic mice. So she did single-cell sequencing. She took punches of the SCN at different times of the day and the night, okay? Then just did this, basically went through standard 10x sequencing, and then with the help of a talented student, they did Surratt analysis to sort of identify subclusters of cells in the SCN, and then specifically neuronal cells. So this is a feature plot, for example, and most of you are familiar, where they use canonical genes from a previously published paper, recently published paper, where they use canonical gene markers to make plots, cluster maps, basically, of cells within the SCN. They also made plots for the EGFP cells here. And then they went back and made more detailed UMAP plots to identify these subsets of cells. So you have 18 clusters here. We have our neuronal cells here, and then various other cells within that cluster. And then they went and did subclustering of the neuronal cells, because that's what we were interested in, right? So you can see up here in this UMAP plot, we got about 7 subclusters of different cells with their canonical, so where they were expressing various neuropeptides, classical neuropeptides seen in SCN cells. And you can see here, they're also plotted those cells that express EGFP, okay? So typically expressing per rhythms. And 2 main cells came up with this expression of fluorescence, okay? EGFP fluorescence here. So these are data of all the time points combined, okay? And then on the right here, they made plots basically of the cells taken at the different time points. So remember, we're looking at circadian changes in day and night, or diurnal changes. So you can see these plots here over 6 time points, all right, day and night. They basically plotted the maps, and here at the bottom, you can see the expression of the EGFP. And what you can see is it changes over time, right? EGFP expression goes up generally at ZT6, right, and then goes back down, right, which is the standard general profile of per expression going up middle of the day and then going down again at night, okay? Right, so this is interesting. So we also, they also did this violin plot, and another way of just visualizing, what's going on. So we have now, these 7 subclusters, again. This is another way of visualizing the data I showed earlier, right? So this is looking at the neuropeptides expression of these cells, and then this is looking at clock gene expression, okay, in the same clusters. One thing you're probably wondering is why PER seems non-existent here. Again, this is data of all combined time points. It's because per expression oscillates as a function of the day, right? So we're just diluting out the effect when we combine the whole data, I believe. And again, this correlates very well with the previous published work that I mentioned, where they use the canonical genes to make these plots. And then Prem also looked at expression, just to sort of verify or look at the core clock gene expression over the different times in the different clusters, right? And for simplicity's sake, she just labeled the green line here, the EGFP expressing cells, and then the black ones are the non-EGFP expressing cells. So one of the first things that comes out immediately is the heterogeneity of it, right? So we knew sort of from molecular labeling that the cells are heterogeneous, but we can see that their rhythmic profiling is also heterogeneous, right? So they're not all having the same rhythmic, exactly the same profile. Well, clock does, but PER1 doesn't, for example, cry and PER2. So main take-home point here is that you get some subsets of cells exhibit very different, relatively different rhythms throughout the 24-hour period. Prem also sort of started looking at, are there certain other genes coming out in these cells that may explain this, trying to think, explain these differences in firing between these cells, between the EGFP and non-EGFP cells. And these two genes came out in her analysis. So okay, so it's a mouthful. Let's just call it protein phosphatase 1 inhibitor. This came out extremely rhythmic, at least in this subset of cells, where they're low ZT2, then they go up, and then go back down throughout the day. Whereas you have this opposing expression, the phosphodiesterase PDE10A. Okay, so that's high in the ZT2, and then starts going down from late day to night. Okay, so this is interesting because recall the physiological firing data, where ZT2 and ZT4 had 6 had very different firing characteristics, right, between the EGFP cells and non-EGFP cells. So she wondered whether these may play a role in this difference in physiological dynamics. So for that, one of the things, and this is actually all pretty much ongoing work, and we are starting to do some, going to start to do some of these recordings. We had some immunoblots here, for example, where at ZT6 and ZT10, just as a proof of concept, she showed PPP1R1, a protein expression level oscillates as a function of day and night, so it starts going up from ZT6 to ZT10. We're doing some behavior, which I'll show in the next slide. And the thing we really want to look at is like the functional changes. Can we, for example, silence PPP1R1 and change its firing state? So if PPP1R1 is responsible for very depolarized state, can we then silence it and make it fire more like a ZT2 and so on, okay? And this is some of the behavior data. So basically, she crossed this PP1 heterozygote knockout mice. Then, in order to get some homozygote knockouts, did behavior. So Samir had mentioned wheel running. We are using this PRR sensors, right? Because wheel running it's very rewarding, but we want to get more naturalistic activity, right? So we can monitor their activity in their cage, and this is just bar just shows dark and light. So we had them in light-dark cycle, and then we gave a light pulse, and then we kept them in constant darkness for the rest of the time in order to see what the degree of phase shift was, okay? This is, again, I'm more happy to, for time I won't go through the detail, but if anyone's interested, please, I'll be happy to talk to you after. And basically what you do is if you give a light pulse to an animal in dark, they will then start shifting, okay? Or you'll induce a phase shift. So long story short, we didn't see a difference between our knockout mice and our wild type. So behaviorally, at least so far, we don't see an effect, right? But of course, there could be other compensatory mechanisms going on, don't know. Yeah, so that's where we are with the behavior. This is something that's ongoing. We will do, so she's going to get knockout, so cross the PP1 knockout homozygous with the EGFP, so we knock out the protein phosphatase 1 inhibitor in these EGFP cells, and then either overexpress or knock down PDE, and then again look at physiological firing. So at ZT6, the cells can be changed, the standard firing at ZT6 down to ZT2 and vice versa, right? So that's something that we are going to start when she comes back from maternity leave. And this is the kind of putative model that we have right now. Again, it's very basic and it's very early, right? So like most models, will probably be proved wrong, but this is the working model. So recall I said for the EGFP cells, they have at ZT2, they are more hyperpolarized and have this very nice firing, relatively hyperpolarized, right? And one way this could be is that ZT2, this protein phosphatase inhibitor, is decreased. This removes its inhibitory effect on the protein phosphatase 1, okay? Now, this then allows protein phosphatase 1 to dephosphorylate, for example, clock proteins, and it's been reported, Uri Albrecht, for example, had published some papers showing PP1 effects on clock proteins. It could also dephosphorylate ion channels, which could lead to these more hyperpolarized state. Because, for example, certain calcium-activated potassium channels are more active when they're dephosphorylated, right? So this would lead to a relatively more hyperpolarized state, for example. It could be that leads to more firing. Conversely, at ZT6, we have this more depolarized state, extremely depolarized, with this probably depolarized block here, but there's no firing. So at ZT6, we know that PDE levels are low, right? PDE typically hydrolyzes cyclic AMP, but when PDE levels are low, you've got cyclic AMP can go and activate protein kinase A, which can go and then phosphorylate the protein phosphatase inhibitor. This makes it more active, right? This can then inhibit the protein phosphatase, which will then allow more of the clock proteins to remain phosphorylated and more of the ion channels to be phosphorylated. And of course, I just mentioned the calcium-activated potassium channels, right? So these then will be less, their kinetics will be actually slower, right? This could be one of the reasons why the cells are more depolarized, okay? Now, to say, we need to test some of this, right? So this is just kind of trying to develop a model and then try and see, do any of these then pathways actually really influence this difference in this dynamics between the EGFP and non-EGFP cells. And yes, so this is it. So this is Priyam, who did all the cool work, and together with Marco, who's a talented undergrad student here, looking for a PhD position if anyone's interested. He's seriously talented. Ching Zhu, who used to be here, now at KAUST, he developed some of the vectors that we started using, but this is a very initial work. And that's Justin. I think most of you know him. We went to Oman for a hike, Samir, which you missed. But yeah, and also I'd like to thank core facility members, Rashid, Mark, Meha, and Nizar for their help, helping Priyam for her work. That's it. Thank you.